Today we have Yves Pommier as our first speaker. He got his MD and Ph.D. at the University of Paris, and then he came to NIH. So he's a chief of the development or therapeutics branch, and he plays a role in the NCI Developmental Therapeutics Program. He's on several other committees as well. And he serves as an editor in cancer research. He also has done several Gordon conferences. He received the Par Ehrlich Award from the Society de Chimie, and he's going to talk to us today about topoisomerase. Yves.
Okay, thank you. All right, thank you, Terry. Thank you, everybody. So this is not limited to one disease because we're talking about enzymes, and we're talking about ubiquitous enzymes. So these are called the DNA topoisomerases enzymes. There are several reviews that you could go back to, and a number of the slides are from these reviews. So these are probably more the drug-oriented aspect of the topoisomerases, and the fact they're very important for medicine because they're used, the topoisomerase inhibitors are used to treat cancer, and they are used to treat bacterial infections, including tuberculosis. And I think this is still an ongoing project. There's still drugs being developed. And the most recent review, which I think now has a large number of citations, is the one we wrote with John Nittis, who is at University of Chicago. So John and I were started in the field many, many years ago, when the field was started by Jim Wang with the discoveries of the first topoisomerases, and then by Jim Shampoo. And that was soon after the discovery of the structure of DNA. Because when it was found DNA was helical, it was clear that that raised a topological problem, and there had to be enzymes to deal with it, and the topoisomerases were discovered. There is also a book that you could find in the library on all the topoisomerases and cancer. So there are different aspects of that. So this is in the library. I think it's online as well. So why do cells have topoisomerases, and what do they do? The issue with DNA is because it's helical, it's some kind of rigid polymer, and DNA tends to make supercoils, so the strands tend to wrap around each other. That's what we call supercoils. And when the DNA is relaxed, so if you take a double-stranded DNA, when you open it up, then you generate on the flank of the bubble that you're opening supercoils, if anybody holds the ends. So on one side, you get positive supercoil, and on the other side, negative supercoil. And the topoisomerases dissipate these supercoils by different ways, but all the topoisomerases do that. The Topo1 mitochondrial, Topo2 alpha, Topo2 beta, they do it everywhere. And then the Topo3s mostly is in negatively supercoiled DNA. The other reactions that they carry out is to decatenate DNA, because when DNA is replicated, if you replicate a circle, if it is supercoiled, you will end up with a catenane. The two circles are going to be intertwined. So in bacteria, in viruses, that happens. In human chromatin, because you have loops, when the loops are duplicated, they tend to be intertwined. So you need to separate, so you need to open one duplex and let the other strand pass through and reclose, and that's decatenation. You could catenate. DNA makes knots. That's been established. It's very clear it makes knots. And the enzyme can unknot DNA by passing one duplex through the other, so it's unknotting, but it can also knot. It's not unlikely the genome, it's known, it has knots. And this may be like on a rope of when you go sailing, the knots are used to make particular pose in marks, and that's probably used also in human chromatin. Also, other structures may emerge when you have what we call holiday junctions, when two duplexes are getting intertwined during recombination, so that's Topo3 alpha. And then you have R loops in the genome and D loops, and this could be also resolved by the Topo3. So Topo3 beta, for example, can do that, and it can also unknot single-stranded RNA when the RNA makes knots. Topo3b can do that. So there's a very broad range of activity of these enzymes. So if you take a human cell, there are 6 enzymes. And the nomenclature is good to know because that's the 1A enzyme are the Topo3 alpha and Topo3 beta. The 1B enzyme are 1 mitochondrial. So the Topo1 enzyme cleave one strand at a time. That's the easy part to remember. Type 1 enzyme cleave one strand. So Topo1 and Topo3 cleave one strand of DNA or RNA for Topo3 at a time. The type 2 cleaves the duplex of DNA, so that's Topo2 alpha and Topo2 beta. And humans have 6 enzymes, 6 different genes to deal with all the DNA complexity. In E. coli, it's a little more simple. They don't have the 1B, so the idea is the 1B probably emerged from a virus that infected the early cells and then was hijacked to relax DNA. But E. coli mostly has 1A, Topo1, Topo3, and it has 2 Topo2As. It has gyrase and Topo4. So Topo1 and Topo1-MT, especially Topo1, is the anti-cancer target of widely used anti-cancer drugs, the camptothecins, topotecan, irinotecan, and drugs that we have developed, andinoisoquinoline. Topo2 alpha and beta are the anti-cancer target of very widely used anti-cancer drugs, etoposide, doxorubicin, mitoxantrone, and gyrase and Topo4 are the target of very widely used and very precious antibacterial drugs, which are the quinolones. And they don't cross over. So the antibacterial do not poison the host enzymes, but the Topo2 inhibitors do not poison the bacteria. So they are different drugs. So if you think of the Topo, what I said, the Topo1 and Topo3. So all the odd numbers are single-strand breaking enzymes. So you have DNA duplex, the enzyme will bind DNA, make a nick, single-strand break, nick, and the nick is then used to relax DNA. And the drugs that are specific for each class bind in this nicked intermediate, which is we call the cleavage complex. So these are the cleavage complexes. This is the name that's being given. So we call them top 1cc, top 2cc, top 3cc. It means there's those cleavage intermediates. And the etoposide doxorubicin poison the Topo2, not Topo1. Camptothecin poison Topo1, not Topo2. And there is no inhibitor for the Topo3. And again, there are 6 enzymes. This is our molecular size. And the Topo2 also works as homodimers. So they're very large protein homodimers, Topo2 alpha, Topo2 beta. Now, if you take a human cell, there are 2 kinds of genomes. There's the nuclear genome, nuclear DNA, and the mitochondrial DNA. Depending on the cells, the amount of mitochondria, this could be up to 5% of the cellular genome is mitochondrial DNA. The topos are distributed in both compartments. Topo1 is specific for the nuclear genome, Topo3 beta for the nuclear genome, and Topo1-MT, mitochondrial Topo1, is specific for the mitochondrial genome. So for Topo1, there is a clear division of labor, Top1 nuclear, Top1-MT mitochondrial. But then for the Top2 alpha, Top2 beta, Top3 alpha, they go in both. And also both genomes have their own repair enzymes, and I will describe that at the end. So let's take them one at a time. So we'll start with the Topo1, then go to Topo2, then Topo3 and the repair. So Topo1, I've told you there are 2, Topo1 nuclear. So when we discovered Top1-MT in about year 2000, we decided not to change the nomenclature and not to call Topo1 Top1 alpha, and the one we discovered Top1 beta, we decided to call it MT, mitochondrial, because it is specific for mitochondria. So as I said, the differences between the Topo1 and Topo2 are major. They are both called topoisomerases, but they're very different. Topo1 works as a monomer, Topo2 as a dimer. Topo1 makes a single strand break, Topo2 makes a double strand break, which is staggered by 4 base pairs. The breakage of the DNA has in common that the enzyme uses a tyrosine. You see for Topo1, this is a tyrosine residue to attack the DNA backbone. And for Topo2, it's also a tyrosine, it attacks the DNA backbone. But the polarity is different. In the case of Topo1, it links to the 3' end, like recombinase, whereas in Topo2, it links to the 5' end, like the Topo3. So this is probably the most ancient canonical mechanism for topoisomerase-induced cleavage of the backbone. So the way the topoisomerases work is by cleaving the backbone and enabling another strand to pass through the broken backbone. Topo1 doesn't use ATP, doesn't use divalent metal, works at 0 degrees, and has its own inhibitors. Topo2 consumes ATP, requires magnesium, doesn't work at 0 degree, and has its own set of inhibitors. So Topo1, which is the DNA untwisting enzyme, if you want to call it this way, there's crystal structure which shows that the enzyme binds DNA. Then after it's bound to DNA, it makes a covalent bond to one of the ends of the DNA, and then the other strand rotates inside the enzyme. So it's a controlled rotation that dissipates the supercoil. Then it realigns, and then the DNA religates. There is no ligase. Everything is done inside the Topo1, and that relaxes DNA. So Topo1 is essential for transcription and replication because it dissipates the supercoils and potentially for DNA repair. Yet in yeast, the knockout of Topo1 is viable. It's compensated by the Topo2 and Topo3. But in mouse cells, human cells, Topo1 is essential. So what I described at the beginning, this idea that when DNA is being transcribed or replicated, the strands are being separated by a helicase, what happens when you have a nuclear matrix attachment, you generate positive supercoil on one side, negative supercoil in the back. For the movement, you need to dissipate these supercoils, otherwise you cannot move along the DNA duplex. And this dissipation is mostly done by Topo1. So Topo1 would bind in these helical domains, superhelical domains, and would make these nicks and enable the shriveling. So it forms cleavage complexes. Normally, as we speak, you're doing it all the time. Every cell is relaxing the DNA. Nothing happens. These cleavage complexes are invisible, except when there is a drug, like a Topo1 inhibitor camptothecin given to a cancer patient, or when the DNA is damaged, then the religation is not proper. And also during apoptosis, Top1 cleavage complexes can form. And this is important because these cleavage complexes, in addition to drugs, can be induced by endogenous and exogenous DNA lesions, some very frequent, a basic site oxidized base, carcinogenic adducts. And this is the reason that in some neurological syndrome, when there are DNA repair deficiency for the Topo1 cleavage complexes, that leads to neurological syndrome because neurons used by transcription, they need to repair. They fail occasionally when the DNA is altered, and repair is essential. So the two Topo1s, the nuclear Topo1 and the mitochondrial Topo1, mostly differ by the N-terminus. They are encoded in two different chromosomes. The nuclear Topo1, Top1 has a nuclear localization sequence, the Top1MT, which emerge in the vertebrates by duplication. Yeast has only one Topo1, but in vertebrates there are two, and it has a mitochondrial targeting sequence, very short but very efficient in the N-terminus. And in that sense, the mitochondrial Top1 really looks like the viral Topo1, like if you take the vaccinia Topo1. It's a very short, truncated version. It almost looks like the mitochondrial Topo1 of the human cells. So the Topo-1s have their own specific inhibitors. They were discovered by the NCI out of a screen of plant extracts in the 1970s, where the NCI was looking for new cures for cancer, new drugs, natural products, and found this very active, potent toxin from this plant, Camptotheca acuminata. This was purified, tested, and found to be due to the presence of this chemical in the plant, which was called camptothecin. This camptothecin has alpha-hydroxylactone, and it's also very unstable. Today, there are two derivatives at least that are used clinically at the clinical center everywhere in the world that are camptothecin derivatives, topotecan, and they have been modified on this region to make them water soluble because camptothecin is very insoluble. And then the second is irinotecan, which is a prodrug. The active metabolite is SN38, and there are other camptothecin in other parts of the world. Now, how do they block the Topo-1? We discovered that some years ago, and that led us to discover a paradigm for a drug mechanism of action. Camptothecin is a planar molecule. As you see, it almost looks like a base pair, but does not intercalate into DNA. And when we looked, so this is a topo cleavage complexes, which normally goes back and forth. When you put camptothecin, even with a small oligonucleotide, you get breaks. And what we proposed is camptothecin was binding at the interface here. And that was based on DNA sequence analysis at the time. And it took about 10 years to confirm that camptothecin, this is topotecan, the camptothecin derivative. In gray is the Topo-1, in green is the DNA. This is the break. And you could see the drug beautifully inside the cleavage complex. So this is what we call a ternary complex. The drug doesn't bind to Topo-1 alone, doesn't bind to DNA alone, but nature has selected that drug to bind only in that particular pocket. And when it binds, the topo is sequestered into this cleavage complex intermediate. It can still eventually go back, but once the drug is in it, it will actually poison this. So we call that the interfacial inhibition because the drug binds the interface of Topo-1 and DNA. Another point that came very early, for you it's probably very early in your life, 1988, when the camptothecins were looked at before they were approved in 2000, it was noted that in yeast the camptothecin were inactive. And what was found, instead of saying that yeast could not take up camptothecin, is that by doing mutants of homologous recombination, RAD52 mutant, they were all sensitive to camptothecin. Moreover, if you knock out Topo-1 in those, they become resistant again. This was confirmed in an independent paper in 1988, and that showed very clearly that homologous recombination is a key pathway for the survival to camptothecin. And to me, historically, this says that the camptothecin were the first drugs showing synthetic lethality in homologous recombination deficient cells. Now, you're well aware that this label is given to the PARP inhibitors, but historically, it was first discovered for the camptothecin, 1988, non-2012. We developed new Topo-1 inhibitors, which are used now in the clinical centers. Because the camptothecin were effective, and Topo-1 was validated, and because we knew that among drug class, if you take different chemical class, they usually have different anti-cancer activity. For example, colchicine, which is a tubulin inhibitor, is used to treat gout. Vinblastine is used to treat cancer. But at the molecular level, they work the same, but chemically, they look different. So the idea, maybe we should look for other chemicals. And also, we knew the camptothecin had limitations. They had especially intestinal toxicity for irinotecan. They were drug efflux substrate, and they were chemically unstable. So, and the chemical instability is what I told you. The camptothecin active form is the alpha-hydroxylactone. In the blood, within minutes, it's converted to carboxylate, and the carboxylate binds serum albumin. So also the camptothecin bind very transiently to the cleavage complexes. And then we developed our own Topo-1 inhibitors. And the development now is being better understood in the context of the fact that the camptothecin are selective for BRCA-deficient tumors. So if we find new Topo1 inhibitor, maybe we could give to those patients who are not responding to PARP inhibitor. And the camptothecin were the only class, and they had their limitations, which I've given you. So the drugs that we've made and that have gone to the clinic now were made with a colleague, Mark Cushman at Purdue University. We started this effort in 1998, and it took many, many years to get the drug to the clinic, but now they're in the clinic. So one is called Andotecan, LMP400, Adimitecan, LMP776, and LMP744. These drugs, recently, actually, about a year ago, we released the data where we tested all these drugs in the comparative trial consortium of the NCI. So these are veterinary clinics all around the United States, and the owner of dogs who have dogs having cancers can bring the pets to the clinic and then get an experimental treatment for no cost. We decided to look at these drugs in dogs with tumors, and we compared 3 of our inhibitors, and we did in lymphoma. The idea was to compare the activity of the 3 drugs, to do full pharmacokinetics, which is easier in dogs than humans, and look at target engagement, gamma H2X, and Topo1 downregulation. Did this with Jim Dorosho, who is the translation director for the NCI, Translation Research, and Emile Leblanc, who is running all the veterinary trials. And it turned out these are survival a tumor response plot in all the dogs. Each bar is a dog. And if it goes under, it means you get activity. And the 3 compounds had activity in most of the dog. And this one was the best compound, actually, in the trial, which was initially not put in the clinic. So at this stage, what we found is that the 2 clinical compounds were indeed active in dogs. And therefore, that pushes for the phase 2 clinical trial. And the 3rd compound was even better. The dose-limiting toxicity was bone marrow, which is expected for chemotherapy, and there was no diarrhea. And that's a big deal because irinotecan has this problem. And the pharmacokinetic was favorable with long half-life. This drug, which was very active, shows tumor retention, and then we could show target engagement. So having done this so far with the Topo1 inhibitor, the next question becomes, especially today, when you start a phase 2 clinical trial, is you have to select for patients who are susceptible to respond. And you have to measure something that will make a hypothesis. And the idea is to have a molecular signature. Who will respond? So I've told you already that HRD, BRCA-type patient, should respond. And the other thing that we discovered is a determinant response, which is Schlafen 11, which is a gene expression marker, which is now being implemented in the clinic. So you have to minimize the side effects. So we're doing targeted delivery. So the next phase for the Topo1 inhibitors, which has been done already for the camptothecin, you could see here the payloads, they are coupled with antibodies. And the most promising compound, which is from Daiichi Sankyo in Japan, is a camptothecin derivative coupled with a HER2 antibody. And the responses are very, very good. And it's very likely the drug is going to be FDA approved by the end of the year or very early next year. So we're putting our own drugs onto that particular approach. So Topo2. There are 2 Topo2s, Topo2 alpha and Topo2 beta. I tend to call them Top2A, Top2B. It's easier to write. The genes are called Top2A and Top2B. So easy to say protein Top2A, Top2B. The division of labor is that Top2A is primarily involved in replication, replicating DNA, relaxing supercoils when they arise, when DNA strands duplex cross during replication, replicating cells. Top2B is mostly involved in transcription and regulating promoters and promoters enhancers. So this is transcription, non-replication. So these are the shape of the 2 enzymes. They're very similar. Their catalytic tyrosine is in the middle. The alpha and beta are most variable in the C terminus. The all this part is very common. And if you compare to the bacterial counterparts, they're also quite similar, except bacteria use 4 genes. It's a tetramer instead of a dimer. Human is dimer 2 alpha, dimer 2 beta, whereas in bacteria you have 2 jars, jar B, 2 jar A, so it's a tetramer. But the machinery is the same. We know the enzymology very well. So the reactions, Topo 2 doesn't work like Topo 1. Topo 2 takes 2 duplexes. So you have 1 duplex of DNA, another duplex of DNA. And what Topo 2 will do is will make my arm go under the other one, but through my arm. I'm not going to go do this. It's going to break my arm, and it's going to put it back, and then my other arm is there. How does it do this? So you have 2 duplexes. You have the gate and the T, transport strand. So the enzyme will first engage one of the duplexes, put the one on the top, close a little gate, open that other strand, make it go through. Once it's through, it religates behind and then releases, and that burns ATP. So what does it remind you of? This is the NIH gate. When you go to NIH, you have 2 doors. This is exactly what you do with topoisomerase. You stay in the middle of the 2 gate for a while, and then you go to the next. That's how Topo 2 relaxes DNA. It's a gated system, at least 2 gates, maybe 3, because in the middle it breaks. This is very safe. This way the DNA never let go, and the double strand break is never visible because it's inside the Topo 2. This magical reaction can decatenate and catenate, can take knots away or put them in, can relax DNA supercoiling when 2 strands. It's a rithase, so the rithe is when 2 strands pass on top of each other. And it can also induce negative supercoiling for gyrase. As I've told you, this double-strand DNA looping around and coming together is very reminiscent of what happens during transcription when you have the enhancer with the promoters, and the DNA makes a big loop. And we now know very well that Topo 2b is very close to these regions, not only relaxing DNA, but probably right at the promoter. So there's a lot of activity, especially in neurons and in immune cells, to know how genes are turned on and off and how Topo 2 beta is part of this particular gymnastic. In replication, Topo 1 will relax supercoils very well. Topo 2 could do it too, but Topo 2 needs a duplex. Topo 1 doesn't need the DNA to be intertwined. It just needs tension. It would just relax it. Topo 2 needs to pass one strand through the other. When DNA is replicated, sometimes you get post-replication catenanes. That's only Topo 2 could do that. And if you have converging forks, you have also that. And I've told you already decatenation. As far as the drugs that target Topo 2, so for the human, there are a flurry of drugs. And they're very widely used. There is the etoposide-type drugs. Doxorubicin, daunorubicin are very strong, potent Topo 2 inhibitors. And there are a number of other drugs, and mitochondrial. And there are catalytic inhibitors that are shown here. For the bacterial Topo 2, you have all the antibiotics that you have heard about. Norfloxacin, ciprofloxacin, gemofloxacin. These are the quinolone bacterial specific, but they work in the same way. By this, I mean it's the same interfacial inhibition. This is a crystal structure of Topo 2. It's a homodimer. The DNA cleaved strand is here inside the enzyme. And if you look from the top here, you see, and through the enzyme, you see the DNA. And the way the drug poisons the Topo 2 is by binding at the interface of the Topo 2 and the DNA. So again, you're getting the same principle of an interfacial inhibition. Antibacterial is the same principle. This is gyrase, so you have 4 subunits, same kind of structures. You could see very similar. Look from the top, and here is a quinolone. It's again bound at the cleavage site. And what the drugs do, they bind at the cleavage site and interfere with the religation, makes cleavage complexes. So I think this leads to this very prominent mechanism that was, I think, first seen for the topoisomerase inhibitors, but I think it exists for many, many macromolecules in the body. Protein interaction could be jammed by natural products like that. So these are the type of interfacial inhibitions for the camptothecin, the adenoisoquinoline. It's at the cleavage site, and the drug specificity is driven by specific hydrogen bonds to the peptide itself, and for Topo2 as well. So the selectivity of these drugs for their particular enzyme is linked not only to the DNA stacking inside the break site, but also to specific interaction properly positioned for each drug and for each enzyme. Topo3. So again, we get two Topo3, some kind of easy to remember, A, replication, B, transcription, is the same as the Topo2. So Topo3 alpha, encoded by Topo3A, Topo3 beta, encoded by Topo3B. Topo3 alpha primarily involved in replication, and it needs to have single-stranded region to function. So that happens in hemicatenanes, and it prevents recombination. So cells that are deficient in Topo3A, which is coupled with the Bloom helicase, are usually hyper-recombinogenic. So Topo3 removes these hemicatenanes and removes the holiday junction. Topo3B is an ongoing work. It resolves R loops and potentially D loops, but it's the only RNA topoisomerase in human cells. There is an RNA topoisomerase. It's not widely known yet, although it's well known in the community, but not to the wide community. So this is the only RNA topoisomerase for human cells and rodent and so on. So why would you use a Topo2 versus a Topo3 during replication? So if you take the Topo2 pathway, what you could do is decatenate all these twisted molecules, and you could separate the two replicated circles. But sometimes you could get at the end a sort of you see this little loop structure, and Topo2 doesn't recognize this because it's single-stranded. And that's where you need Topo3 alpha, which works together with Bloom helicase. And it's reminiscent in bacteria, they have the same system. It's Topo1A with RecQ, and in yeast, it's Topo1 RecQ. So in replication, Topo3 alpha is always needed. Now, what about Topo3 beta? It was forgotten for many years, and in 2013, it woke up with two papers showing that in a group of patients in Finland, had high prevalence of mental retardation and neurological disease. When they sequenced the patient, they had a deletion of the Topo3 beta locus. And what turned out is Topo3 beta is actually very important for transcription, and it's very important as an RNA topoisomerase. So this was done by Weidong Wang, who is in Baltimore at the Aging Institute, and this was done by a larger group outside. And Weidong is the pioneer, still working very much on this. We also have the Topo3 beta knockout mice, which are viable, which is a surprise, but they are viable in spite of the importance of the enzyme. So how do these Topo3 work? They don't really work alone. They usually work in conjunction with a helicase in the case of Topo3B, like Bloom. And it has a scaffolding protein. And in the case of Bloom and Topo3 alpha is RMI. In the case of Topo3 beta, it's TDRD3. So those of you who study transcription and RNA metabolism, you know about fragile mental alteration protein. This is linked to TDRD3, and that's linked to Topo3B. So it's making a connection with neurological disease and Topo3 beta as we move along. So I think it leads to the question of topoisomerases, genomic integrity, and human diseases. Abnormalities of topoisomerases, lack of, overexpression of, or lack of repair of topo-CCs leads to genomic instability. And it's becoming more and more obvious that this is happening in patients. Because the topoisomerase-induced DNA damage is such that when you have a cleavage complex on DNA and replication collides, you generate double-strand ends. And that's very lethal if you have cleavage complexes. If a Topo2 CC is severed, it would make a double-strand break. This doesn't happen. It's as if you're trying to break the DNA gate. I think you'll need a big truck to do that, so it needs a lot of energy. But it may happen that Topo2 breaks may sever. Polymerase and helicase can collide. And then in some case, you leave behind some supercoils or knots or catenanes, and this is disaster at mitosis. In the review 2016 that I mentioned at the beginning in the Nature reviews, we listed here the number of causes of alterations of, that create topo cleavage complexes and the list of enzymes that do that. So I've mentioned already that for Topo1, one way the topo cleavage complexes, let's say in your neuron, you have oxidative damage, Topo is stuck, and it's not repaired quickly by its repair enzyme. What would happen? Well, if there is transcription, it will collide, and then it will form a permanent cleavage complex. If it happens in a replicating cell, the replication fork would collide. You make a double strand and a single-ended double-strand break, and that is irreversible. And cells have different ways to deal with that. And when they don't, you get human disease. So for Topo1, so there are neurological disease due to lack of removal of Top1cc. For Topo2 beta, which I mentioned is associated with transcription, chromosomal translocation happen at Topo2 BCC. So secondary leukemia of patients treated with etoposide, for example, are thought to be due to translocation arising from Topo2 cleavage complexes that have not been repaired. Topo3b, I've mentioned, neurological disorders. And then there are two repair enzymes for the topo cleavage complexes that leads to ataxia and neuropathy, TDP1 and TDP2. So I will end on telling you a little bit about these enzymes. So the topo cleavage complexes, as I mentioned, go through a covalent intermediate. You see it here for the Topo1 and Topo1-MT. You get 3' phosphotyrosine linkage. For Topo2 and Topo3, you get a 5' tyrosine linkage. Normally, this reaction is reversible because here the 5' hydroxyl can eliminate the phosphate, and here the 3'. But if it gets stuck, you get a cleavage complexes with a covalent topo cleavage complex, a break, and a covalent DNA protein crosslink. How is this removed? So this is a scheme of these drawn different ways. So this is a Topo1, for instance, covalent crosslink. See, this is sort of the DNA backbone. You could see the covalent linkage. Let's assume this is not reversible. We now know cells have two main pathways to remove these lesions. One is a surgical pathway that with a phosphodiesterase. So these enzymes are called tyrosyl DNA phosphodiesterase. TDP1 repairs Topo1 cleavage complexes, so it can excise them. For Topo2, see the polarity is opposite. It's at the 5' tyrosyl linkage. The tyrosyl DNA phosphodiesterase is TDP2, and then it will release the Topo, then DNA repair can take place. But there is backup, because this is probably happening so often that cells have many ways to deal with it beyond the TDPs. They use nuclease. And the nuclease will chop the DNA with the Topo and release the thing and do a little gap feeding. So that's true for the Topo1 and for Topo2. And these nucleases are nucleases you have heard, they're very prominent, like MRE11 or MUS81. And we now know that in BRCA deficient tumors, MRE11 is very deficient. So there is lack of repair of these Topo2 lesions. And one hypothesis has been made is in breast cancer, the prevalence of BRCA deficiency is because BRCA is not helping MRE11 to remove Topo2 cleavage complexes. So the idea of promoters driven by estrogen, you get a lot of Topo2 cleavage complexes. These would happen in every, especially highly replicative mammary cells during the menstrual cycle, or during lactation. So they are always removed. But if they are not removed, and if BRCA needs to help to bring the nuclease to these lesions. And one of the mechanisms proposed for the selectivity for ovarian and breast cancers is that BRCA-ness is BRCA cannot drive the repair of top2 CCs. So schematically, you could think it this way. The two main ways to deal with this is the phosphodiesterase pathway. So if it's a Topo1, it's TDP1. A Topo2 is TDP2. If the enzymes are not efficient, that leads to neurological syndrome mostly. If the endonucleases are not efficient, CTIP, MR11, MAD50, NBS1, or some of the XP, then you also get in problem. So these two TDPs have different activity even beyond the topoisomerase repair. So they were discovered because of the topoisomerases, but it turns out that the TDP1 has a much broader range of DNA repair functions. So this is also not so well established, not so well acknowledged, I would say, in the DNA repair field. But this repairs a lot of lesions by alkylating agents, blocking lesion at the 3' end of replicating DNA. It works in mitochondria in the nucleus. As I've told you, TDP1 deficiency is found in patients who have a nervous system degenerescence, and it's coupled with PARP1. TDP2 has a much narrower spectrum of activity. It's mostly Topo2 cleavage complexes, but it is also involved, presumably, during poliovirus replication to excise the tyrosine at the end of the nucleic acid. So normal cells have mainly two pathways to repair these complexes. The tyrosine phosphodiesterase pathway for Topo1, which is coupled with PARP1. Alternative pathway is the endonuclease pathway with the XPF, ERCC1, MR11. And for Topo, and if one of these pathways is deficient, then the cells only rely on the second. So there is a synthetic lethality here, because if you block this pathway with a PARP inhibitor, then the cells become exclusively dependent on the MR11 XPF pathway. And that is a clear synthetic lethality of maybe providing an opportunity to give PARP inhibitors with Topo1 inhibitors in MR11 and ERCC1 deficient cancer. And MR11 mutations are not rare. They are found in mismatch repair colon cancer. It's been underappreciated, but if we took those particular patient tumors that are MR11 deficient and gave them a Topo1 inhibitor plus PARP inhibitor, maybe we would see responses where we don't see them now. So I'd like to make the acknowledgement. A lot of the work I've been doing on the drug side is developing the adenoisoquinoline. And I've already mentioned my colleague at Purdue University, and Jim Dorosho, who is the NCI Director for Translational Research, is also in our branch, and then other colleague at the Developmental Therapeutics Program. So I'd be happy to answer your questions, or if you want to send me an email, it's easy to find my email.
Thank you. Any questions? So how fast do the topoisomerases unwind the DNA?
The speed we calculated with Keir Newman, who is in this building. So with Keir Newman, we're doing single molecule analysis. And my approximation for Topo1 rotation, the rotation inside the enzyme is about 6,000 RPM. So it gives you...
Pretty fast.
Pretty fast. These things work in a second millisecond. So when we do biological experiment in hours and minutes, it's not at the scale of what cells do. Cells work in a millisecond. And the passage, I don't know how quickly a passage, you know, it's like going through the gate of NIH. It takes you a little bit. You have to put your card twice. But I think the Topo2 passage, I don't know the speed actually, but it's probably pretty fast. So these things work very, very efficiently and continuously in your cells, in my cells, working all the time, whether we transcribe or replicate or whatever we do. So these are very essential machines.
And do they alter the cell cycle? Do the cancer cells then build up, say, in G1 phase?
Usually cancer cells overexpress the topoisomerases because they replicate fast, they transcribe a lot. So it's very common, the overexpression of Topo1. In breast cancer, there is amplification with HER2 of the Topo2 alpha, which is near. And that probably enables them to replicate and transcribe quickly. their Achilles heel is when they have a lot of topo. Once you put a topo poison, then they're sensitive to topo poison. So because then you poison the enzyme that the enzyme is using. So it goes both ways. So if a cell has a lot of topo, it should be more sensitive to a topo inhibitor. And that's the case usually. So yeah, cancer cells tend to put a lot of these enzymes up. Yeah.
I was just wondering if you can expand on the relationship between the BRCA mutant breast and ovarian cancers and the synthetic lethal approaches of PARP inhibitors and topoisomerase inhibitors, because I think in my understanding, at least, resistance to PARP inhibitors emerges very quickly, and they're not actually as great in the clinic when treating patients as they are in tissue culture. So I was wondering if you were suggesting that we can use them like sequentially or once resistance emerges, or if it's like, because I think also one of the mechanisms is through RAD51 loss, which then rescues the whole HR defect, and in which case I think topoisomerase inhibitors would maybe not work as well.
Yeah, I think it's you bring a number of questions. So connection with BRCA and Topo2. So this has been worked on last year and the year before, mostly by Shunichi Takeda in Kyoto and André Nussensweg had a couple of papers on this. And the concept, as I said, is that when the cells through estrogen or prostate cells through androgen active the androgen responsive element or estrogen responsive element, they have to recruit Topo2 on the promoter to facilitate transcription. Now, these breaks at the promoters tend to be pretty stable, and the topo cleavage complexes tend to accumulate. And it's a mystery for us why they are so sticky in a way, because usually the topo go away. In those places, they don't go away quickly. And the idea is that the cells use the endonuclease pathway to take them off the hard way. The topo doesn't let go by itself, so they have to take it off. And the idea is the BRCA proteins are recruited to facilitate the recruitment of the nuclease for that. So if you have a BRCA deficiency, the topo CC keep accumulating, they're not repaired. Then in these hormone-dependent tumors, you get the mutations, and then you get eventually translocation, and you get all the bad things. So that's maybe one aspect of your question. The other aspect for the PARP inhibitor, I'm happy you're saying this. Everybody swears by PARP inhibitors. When I first looked at the PARP inhibitors, the most striking finding is when we took the 60 cell line of the NCI60, about 1/2 of them do not respond even to 100 micromolar. So what is it? But some responded at 10 nanomolar. So you're totally right. Some cells are black and white, they either respond or non-respond. So they are not as potent as we would think. And the determinant of response, when we looked carefully, was not their BRCA-ness. It turned out when we looked carefully genomically, it was Schlafen-11, which was unrelated, unknown, and so on. But this is another story. In the case where the PARP inhibitors don't work, even if the tumor is BRCA deficient, because that happens, because as you said, they will compensate by their pathway, then the Topo1 inhibitors may be a way to go. So that's what we're proposing. Or maybe you add a Topo1 inhibitor to a PARP inhibitor. That may limit the resistance that occur. So that's what we're going to be testing, is whether adding a Topo1 inhibitor to a PARP inhibitor will increase the fraction of responders and eventually limit the fraction of patients who escape. For the moment, it's more hypothetical. But clearly, Topo1 inhibitor, the three classes of drugs that are BRCA or HR dependent, PARP inhibitor, yes. Cisplatin, in part, because platin is also NER. It's not purely, so we all want to say it's HR, but it's more complicated. And Topo1 is pretty good HR. So these are the three classes. But the others don't depend. Topo2 inhibitor, no HR, many drugs independent of HR. So these are the three classes I know: PARP inhibitor, platinums, Topo1 inhibitor for BRCA deficient, HR deficient patient. That from the preclinical model, that's clear.
Okay, if there's no further questions, we'll take a 5-minute break. So Kurt Harris, our next speaker, he's chief of the Laboratory of Human Carcinogenesis. He got his MD from Kansas University, and he studied internal medicine at UCLA, Washington Veterans Administration Hospital, and NCI. And then he came to NCI, and he's also an adjunct professor of medicine at Georgetown University. He's won several awards, including the AACR Princess Takamatsu Award, and he's chief of the journal Carcinogenesis, and he's going to talk to us today about lung cancer, precision medicine strategy.
Okay. All right, Terry. So all of you diehards who are still here after 5 o'clock, actually, I found that many of my postdocs are staying after 5 o'clock nowadays. I don't know why. I mean, there was a time, I forget about that. Let's talk about science. Let's talk about lung cancer and precision medicine. So what is precision medicine and when did it start? It started here at the NCI, and Harold Varmus and Francis Collins had a Institute of Medicine group get together and come up with what they call precision medicine. It's essentially an update of the kind of medicine that is ongoing and was ongoing. So traditional medicine at that time was to get a family history and do signs and symptoms and get some laboratory tests, and that's how it went for many, many, many decades, if not centuries. So what's precision medicine? It's multi-layered, it's individual-centric, and it's interconnected. And one of the keys is a information commons, which has these omics, and I've added clinical information and epidemiological data. And this gets put together to form a knowledge network and a new molecular taxonomic classification of individual patients to guide clinical studies and improve clinical investigations, to prevent, to guide prevention research. I added that wasn't in the original one, and to inform biomedical research. And these have feedback loops that enrich and expand the information commons. So lung cancer, I think you heard about a session on lung cancer fairly recently, actually. It's one of the things we study in the Laboratory of Human Carcinogenesis. This is at the time of the 50th anniversary of the Surgeon General report, they did a calculation of the number of cancer-related deaths and various and disease-related deaths, and came up with about over 20 million people died from tobacco use. And of course, that's still ongoing. In the United States, the incidence of is going down in males and is still pretty plateaued in females. Let's see if this works any better. Yeah, that works better. Okay. The data in 2019 or 2020, there'll probably be an update of this, and there may be a slight decrease in female lung cancer by that time. And eventually, male will coincide with the female. So what causes lung cancer? Obviously, tobacco smoke. Why is it going down in the United States? The United States, we used to look like this, and that's about 35 to 60 smoke pack years, or the frequency of smoking, I should say. And that's still quite common in Russia and Asia and a number of other countries. And we used to look like this. And that's why lung cancer was so has been so prevalent in the United States. So the various people in the Laboratory of Human Carcinogenesis, and there are 5 PIs, including myself, we focus on studying the major lethal types of human cancer. Lung cancer, whoops, sorry about that. Lung cancer, and Reed Ryan and myself study it. Xin Wang, liver cancer, breast cancer, and prostate cancer. Stefan Alms and pancreatic cancer is Pervez Hussein. So that's, and we're talking about, it's probably closer to 5 to 6 million deaths a year from just this group of cancers. So a number of years ago, actually in 1981, I participated in a meeting in Greece, and this is Cape Sunion, and this is a watercolor from Professor Hirayama. And Professor Hirayama reported the very first evidence of passive smoking and induction of lung cancer. And I will tell you, of about 400 or 500 people at this meeting, at least half of them didn't believe it. But he proved to be right. So there are probably over 100 papers now in the literature showing that passive smoking that you get in your workplace or from your spouse is associated with increased risk of lung cancer. And I'm rather fond of these old kinds of quotes, and this is from Hippocrates. Some men / women have constitutions that are like wooded mountains running with streams, others like those with poor soil and little water, and still others like land rich in pastures and marshes, and yet others like the bare, dry earth of the plain. It was essentially that people differ from one another. And that was obvious even thousands of years ago in regards to risk of disease and other things. So I was sitting at this meeting, and I said, well, is there any evidence, or could I find some evidence, investigate whether or not smoking of your parents increases your risk of developing lung cancer? So this was in the late 80s, or early 80s, I should say. And women didn't start smoking in the United States heavily until Second World War. And then you don't get cancer until 50, 60, 70 years of age. So I figured it would take about 35 years before I could do that study. But in fact, we did do the study 38 years later. And the hypothesis was that childhood exposure to secondhand smoke and genetic alterations in innate immunity increased lung cancer risk in never smoking adults. And Susan was involved with this, and we collaborated. One of the cohorts came from the Mayo Clinic, one from our cohort at the University of Maryland. And we, in fact, did find that parental secondhand smoke exposure during childhood is associated with dose-dependent increase in lung cancer risk among never smokers in these two different cohorts. And whether it was the mom or the dad or both, if it was mom and dad both, it was additive. There was even some evidence that grandparents taking care of the kids at an early age increase their risk. It was especially true in mannose-binding lectin type 2 haplotype, which is reflective of a high active innate immune system. And surprisingly, these cases developed cancer primarily in their 50s and 60s instead of in the 60s, 70s, like most of us would. So that led me to think about the expososome, and what is that? That is something that was defined by Chris Wild, who was the director of the International Agency for Research on Cancer. And it involved not only the external environment, tobacco smoke, infections, diet, toxins, radiation, but also the internal environment that is related to that. So he added obesity and chronic inflammation, and later on the genome changes and epigenome changes, and development of cancer biomarkers of risk and diagnosis for prevention and screening, and understanding carcinogenesis feeds into this, but it also leads to cancer therapy, as I'm sure all of you know. So genome, well, how about lung cancer in the genome? Well, the traditional view of lung cancer when I was a resident and medical oncology fellow was small cell, adeno, and squamous. And then in '87, KRAS mutations were in fact found. And I will say that P53 mutations were found much before this. And following KRAS, EGFR mutations, and then finally, in about 2014, there have been a couple more that have been found, were primarily ALK fusions and a few other things that are associated, such as BRF1 and MET, with lung cancer. So what causes lung cancer and causes cancer in many situations? There was a period of time which people were studying chemical carcinogenesis, including myself. And you could go epidemiological back into the 18th century, and chimney smoke, sweepers, developed scrotal cancer from benzpyrene primarily. Aspergillus flavus, which I'll say something about more, causes formation of alpha toxin B1 and liver cancer, and then smoking has about 66 different chemical carcinogens in it. So it's a witch's brew of carcinogens, and that's why it's so carcinogenic. So there have been three eras or 3 decades, which I've listed here as examples of initial seminal advances in exposure of environmental carcinogens that are being molecular linked to mutagenesis and to epidemiology for that point. And so back in the 70s, they were finding that carcinogens, chemical carcinogens, are mutagens. So that was all very exciting, but it sort of tied the carcinogens with DNA damage. And then the discovery of P53 mutations linked environmental carcinogen exposure. And the initial studies were done by our group and by Ozturk. So it's 2 different cohorts. And there's an interesting story about that. I won't have time to tell you about that. And there's also a review that we wrote for multiple kinds of carcinogens that cause P53 mutations. So this was sort of the start of molecular epidemiology, and which I and other people contributed to. And then more recently, in 2013, Ludmil Alexandrov and Mike Stratton have published various sequences. They're trinucleotide sequences. Instead of looking at one gene, it's looking at the whole genome, and published in Nature as essentially a link between looking at a single gene and then looking at the entire genome. Before that, it was found that alpha toxin, as I mentioned, causes G to T transversions, particularly at one codon, 279 of P53. Smoking at various hotspots, again, G to T with strand bias. And strand bias, in this case, means that one of the alleles, the wild type allele, since there are 2 alleles, is lost, and then you have mutation in the other. Ultraviolet light has a very characteristic CC to TT transition, which is not found with any other carcinogen that's known. Astrostrolic acid which is causes kidney tumors, is A to T transition. So that was sort of the status of things back in the early 90s, and it went further than that. But P53 goes back 40 years now, when it was first discovered, and then finding mutations in it was discovered after that. There are some cancer types, such as ovarian cancer, in which P53 mutations are almost over 90%. And some of these mutations are loss of function as tumor suppressor, and others are gain of function. So it switches from a tumor suppressor to an oncogene, a Jekyll and Hyde kind of switch, if you want to think of it in that way. Now, not all mutations are equal. Some are purely loss of mutations. Some are with less and less wild-type activity, such as these. Others gain oncogenic activity, and these are just examples of that. So developing therapies against the oncogenic forms of P53 is something that the field has been involved with for about 10 years now, and progress actually is being made, and we'll learn more about that in May in Rehoboth, Israel, when the next P53 meeting is held. Well, P53 and its functions affect multiple levels of the omics data, and this is just a nice cartoon that we put together, Anna Robles and myself, a few years ago. And there's, I mean, why people are still interested in P53, it just does lots of things. All right, now let's go back to this computational analysis that Ludmil and Mike Stratton did. This was their initial paper in which they described 21 different signatures and looking at multiple cancers, including lung cancer and head and neck cancer. And in those, smoking, there was a specific signature that was associated with smoking and those kinds of cancers. There are signatures that are associated with age, so just replication of cells. That's an interesting aspect of cancer and aging. And these are just some other examples, and this has been markedly expanded, so there are at least, I think, over 35 different signatures now that they've characterized. And the aflatoxin one is signature 25, as I recall. There we go. So one of the kinds of things that they have done is compare smokers versus non-smokers. So signature 4, that's the blue lines, are much more common in the smokers than in the never smokers or non-smokers. And they've looked at several others. So those are the kinds of correlations that have been made, and they actually make sense from knowing the various carcinogens that are involved, such as benzpyrene. So this is an example of the kinds of data that they see, C to A transitions, for example, that I mentioned, which is signature number 4. If they took humanized mice, in which it's humanized with human P53, and expose those mice to benzpyrene, one of the prominent carcinogens in tobacco smoke and in polluted air, they get the same sort of signature. Or if they take human embryonic cells, they get the same signature with the exposure. So that's the kind of links that they're making between exposure to various carcinogens and mutagens and human cancer. So this just shows that gain of function mutations, patients have a poor prognosis than those that are wild type versus those that are just loss of mutation. And if you looked at codon 249 in liver cancer, it's a gain of function, but it's like this. So that one mutation in liver cancer is highly deadly, and it's caused by aflatoxin. All right, chronic inflammation has been tied to human cancer back to at least the 1800s. And these are examples of primarily inherited versus acquired hemochromatosis, iron overload, Crohn's disease. You could put other diseases associated. Familiar pancreatitis, which was found in certain families, such as Carter's family, and he's one of the few members of that family who's still alive and didn't get pancreatic cancer, although he has metastatic melanoma now at age, 99, I think. More common are acquired versus inherited, and they can be viruses, bacteria. I'll say something about acetovorax and lung cancer in a minute, and parasitic infections. And the global impact of inflammation is about 2 million cancers per year. It's been calculated or related to infection. And other causes of inflammation are associated with many more cancers per year. So, for example, in addition to have those 66 different chemical carcinogens, smoking is a very potent inflammatory agent. And any of you who took a draw of cigarettes in your early age, hopefully not later, probably started to cough because it's really toxic. And if it wasn't for the nicotine, and the nicotine is, kids are being hooked with nicotine with these cigarettes right now, no one would be smoking, in my opinion. Various inflammatory agents have been investigated. These are some of those that we found. In a prospective study, we found that IL-8 and C-reactive protein is found in European Americans, and in a study that collected samples 20, 30, 40 years ago by the epidemiologist over in Shady Grove now. And so we found those were predictors of developing cancer, actually years prior to developing lung cancer. And at the time cancer was found, IL-8 and IL-6 in both Afro-Americans. There were very few Afro-Americans in that initial study, but the Afro-Americans also have an increase in IL-1 beta and IL-10 compared to the European Americans. So there's some health disparity differences that we and Reed Ryan and others are investigating. And then poor survival. So, and this is poor survival primarily of stage 1 lung cancer. So stage 1 lung cancer is about that big, and there's no metastases or no detectable metastases. And but if the case or your patient, or if you're on the outside, your client has elevated of these different cytokines, or mannose-binding lectin, which I mentioned earlier, you have an increased probability of dying of a recurrence. So now let's look at transcriptome. This will take us back about 2 or 3 decades now, in which Victor and Gary discovered there were non-coding RNAs. There's a non-coding RNA lecture part of the series, isn't there, Terry? Yeah, okay. So you know about all of that. And that was quite interesting at the time. And Carlo Croce decided to look at human cancers, and he found that microRNAs are differently expressed in human cancers, and that they can predict risk diagnosis or prognosis. And most of those studies were done with our group. So as you learned from the non-coding RNA group lessons, the microRNAs can inhibit translation, or if there's a bit of a mismatch, they can cause a message to cleave. But they can also bind to proteins and affect protein function. And they can bind to receptors. increase IL-6 and TNF-alpha. So that's pretty interesting in terms of inflammation. So this is Nozumo when he was a young fellow in the lab. And we found that microRNA profiles were significantly different between primary lung cancers and corresponding non-cancerous tissue and among different histological types. And found that miR21 and these other two were overexpressed and lead-7 was underexpressed and was associated with diagnosis and prognosis, again, of stage 1 lung cancer. And we're primarily interested in stage 1 lung cancer and why they recur. Another study that we did, and this was in collaboration with Carlo, we looked at 6 different cancer types. And at that point, there was, I think, 190, maybe 200, and so microRNAs that have been discovered. Now there's over 2,000. But of those that were discovered at that time, we looked at these 6 different cancer types and found that miR21 was overexpressed in all of them. And when we looked at other kinds of cancers in the field, looked at other cancers, it was upregulated in 18 major cancers and a biomarker poor prognosis in 14. So we figured that was probably pretty interesting and important. And so our group looked at 3 different cohorts, one in Maryland, one in Norway, and a Japanese cohort. And there's some difference here. We use primarily cancer-specific mortality, which I much prefer. The Japanese prefer the relapse-free survival. So their data looks better than our data and the rest of the world. But nevertheless, if miR21 is high, that's the red, poor prognosis in all 3 of these cohorts. And then the same is true in a large number of different kinds of cohorts. So high levels of miR21, poor prognosis in multiple kinds of cancers. So what's the mechanistic underpinning of this? Well, one is that miR21 is amplified in the genome fairly frequently, especially in lung cancer. It leads to decreased transcriptional silencing. We found that if there are EGFR mutations, you don't find KRAS mutations, but you find an increase in miR21. If you find KRAS mutations, you don't find EGFR mutations, and you find an increase in miR21. So they're in the same pathway or a similar pathway. And David Baltimore found that various cytokines increase STAT3, and one of the genes that STAT3 increases is miR21, and different kinds of stress, ionizing radiation, and free radicals increase miR21. So how does it work? Well, it inhibits a variety of different gene expression of different genes. And most of those are ones that are involved in inhibiting cancer in various ways, or in inhibiting mismatch repair. So you have less mismatch repair with increased expression of miR21. So, and there's a lot more data that's been collected since I prepared this slide a number of years ago. So then we went and there was a time in which there were people looking at expression of large numbers of protein coding genes. And we took a different approach. We decided that we wanted to look at a minimum number of large expression of coding genes, including we found in these studies and analysis by random forest and other ways an increase in XPO1, which is involved in transport of microRNAs, BRCA1 expression, high levels, which has been previously shown by others to cause genomic instability. HEF1 alpha, Nobel Prize, that was won this year by 3 different groups, and also decreased DCL1. which is a tumor suppressor gene that was discovered here at the NCI. So we found that this 4-gene classifier was quite reasonable in identifying, again, stage 1 adenocarcinoma in these original 5 cohorts, as you can see. So this is 100% survival, that's 0% survival, and this is time in years. So when we updated that with 7 more cohorts of stage 1 adenocarcinoma of the lung, essentially the same thing was found. And even stage 1A, changes in those 3 genes was associated with poor prognosis in both stage 1A and stage 1B, which is 3 to 5 centimeters in size. And this is just looking at all those data put together. So we decided we wanted to ask the question, could we combine protein coding genes that are mechanistically related to lung adenocarcinoma and non-coding miR-21 to find a better prognostic classifier than either one alone? And that's what we did in the Japanese cohort. Again, this is relapse-free survival versus, so this is a 4-gene, this is the miR-21, 4-gene, miR-21, high in each one of those, poor survival, and if you put the 2 together, poor survival, and especially in primarily 1B versus 1A. So how did we think about that at the time? And what we came up with was non-coding RNA and coding RNAs. If you put them together, if they're both low, if either one is high, or if both are high, again, these are survival Meyer curves. And both can identify people who are at risk, but each misclassifies some patients, but they misclassified other patients. So the combination of the 2 is better than just one. And we found that not only in lung cancer, but in colon cancer, esophageal cancer, both adeno and squamous, in lung cancer, which you've seen, and another group found that to be in breast adenocarcinoma. So that hypothesis actually, or principle, has been found in multiple kinds of cancers now. So let's look at the third omic, and that's the epigenome. And again, we're looking at lung cancer. And this was done by Anna Robles. The hypothesis was that integrated biomarker classification in stage 1 lung cancer, adenocarcinoma, based on independent message, microRNA, and demethylation markers will improve the prognostic classifier. So it's going from 1 to 2, now 3. So one of our colleagues, Manel Esteller, had actually studied DNA methylation in stage 1 lung cancer, and had come up with a signature which included all of these marks, DNA methylation marks. And I looked at that and I said, well, maybe all of those are dependent on each other, or maybe they're independent of each other. We analyzed that. We found that they're all dependent on each other. So we decided, well, let's look at the one that's most highly found, and that's HOXA9. So we went from 5 or 6 down to 1 again. So the objective is to get smaller and smaller. That gives you better and better results. And indeed, if we put all 3 together in stage 1 lung cancer, these are Kaplan-Meier plots again. So this is months, this is a 5-year point. So if all 3 are positive, classifications, omics are positive, very poor prognosis. Stage 1A is a little bit better than stage 1B. So that was something that impressed us, and the NCI has patents on all this stuff too. So now something newer, and that's the microbiome. And was there a lecture on the microbiome? Okay, so should be familiar with this. And the microbiome is about 10 times, 100 times more bacteria and fungi on our body in our gut and elsewhere than there are human cells. And the microbiome varies depending on the surface that they're on or the interior that they're on. And so Julie Segre and others here at the NCI and the microbiome program actually made very important discoveries of this in the variation, and we collaborated with Julie. Well, it was already known a few years ago now that there were bacteria associated with an increased cancer risk, A. pylori, and fusiform bacterium in colon cancer, dysbiosis. That means there's a change in the frequency of different classes of bacteria in cancer therapy. Doug Lowy and John Schneider found a human papilloma virus in the vaccine, and recently we published data on acetovorax, which is a gram-negative bacteria, and it's associated with lung cancer. And all of these have different effects on cells, either on when they're on the surface of a epithelium, for example, or viruses can cause mutations. They can cause problems with the mucosa barrier, especially cigarette will cause this, so the bacteria get in and cause inflammation. And some of the bacteria produce actually proteins, which are mutagens, which is another interesting way of causing mutations in human cancers, in human cells, causing cancer. So in the study that we recently published, I guess sometime last year, we found that there was an interaction between the microbiome, smoking, p53 mutations in human lung cancer. And we found several taxa, including acetovorax, were higher in both adenocarcinoma and squamous cell carcinoma in smokers versus former smokers and versus non-smokers. So former smokers and people who were current smokers had an increased frequency of acetovorax. And a group of these taxa were associated with squamous cell carcinoma, and as I said, were enriched in smokers, and that these were associated with p53 mutation. So was p53 mutations actually playing a role, or was it a consequence of the fact that these were smokers? And that's an interesting kind of question. This came to a conclusion that the squamous cell carcinoma-associated taxa are enriched in tumors with p53 mutation, which establishes a microbiome gene interaction in human lung cancer. And then the next question is, well, are these bacteria passengers, or are they playing a role in the development of the cancer? So we're doing a large number of animal studies and other kinds of studies right now. I'll just show you one slide, maybe. Oh, that got delayed, or I guess it didn't get transferred when he transferred my USB stick. And we have found indeed that acetovorax increases the size of the tumors in a mouse model that Tyler Jacks developed with KRAS and p53 mutations, and that they got tumors quicker than if it was acetovorax, or if they were exposed to sham, or these are intranasal installations, or to lactobacillus, which actually is an anti-inflammatory agent. So it looks like it's related to inflammation, and so we have experiments now looking whether or not uh, inflammatory agents, either chemicals or drugs or lactobacillus, will decrease the frequency of lung cancer caused by acetovorax in this animal model. So let's, I guess we're about ready to end up looking at the metabolome, which was in the previous slide. And the hypothesis that these two postdocs, Maya and Yves, Yves just taken a high-level position in cats, and Maya has a high-level position at FDA now. Unbiased metabolomics will discover biomarkers associated with risk, diagnosis, prognosis, and therapeutic outcome of lung cancer. So let's go back to a previous time, Hippocrates again. So Hippocrates, it's said, tasted urine for diagnosis of disease in his patients. Now, I'm not as old as Hippocrates, although my kids might think I am. But I've never tasted urine, except when we had small kids. I was changing diapers. So that was one of the things that physicians did at those days. And then a physiologist, Armstrong, in Europe, and this is a picture of all this, writes, from a liquid window through which physicians felt they could view the body's inner workings, urine became the beginnings of laboratory medicine. I mean, we're not allowed to write this kind of flowery language, but I think it makes a point. And this Dutch painting is a good example of that. Okay, so we did an unbiased screen initially in urine, chromatography and mass spec analysis, and we found that there were 4 different metabolites, one creatine riboside, one in acyl niromanic acid, the other is cortisone sulfate, and now we've recently identified what that peak was. If you put all 4 together, the prognosis is very poor. 3 out of the 4 also poor, and it gets a little bit better if only 2 or only 1. And also diagnosis. This is a Roc curve, so this is looking at specificity and sensitivity. And so the area in the curve here is, let's see if I can read it here. It's actually not, yeah, it's down there. It's about 0.9, which is actually quite good. And predictive value, positive predictive value, and negative predictive value. Physicians, including myself, look at the negative predictive value because you have someone who has low-dose CT scanning and there's a lesion there. The negative predictive value might allow you to say, well, why don't we wait 3 months and see if that lesion increases or not? Because most of them are not cancer, and I'll show you that in just a moment. So we also found that the amount in the urine was associated with the amount in the tumor, and this is a correlation curve. It's pretty good, and high levels in the urine. It's also the correlation here between urine and tumor also holds up if you look at serum or plasma. So a clinical test is being made of this right now, CLIA-based clinical test for licensing and hopefully put in clinical practice. So let's go to low-dose CT. The turning point here was in old days, that wasn't such a long time ago, chest x-ray was the screen, and you didn't see anything until they got stage 2 or 3 or 4 cancer. But low-dose CT decreased the mortality about 20% in this original study. In the study done in England, it was closer to 30%. And women was better at predicting cancer and decreasing mortality in women than men. So this was a case control study, and I should say, comparing X-ray versus low-dose CT, and the false positive rate was pretty high. I'll make a point of that in a moment. But it did detect primarily early stage 1A and 1B lung cancer, in which the cure rates are the best with surgery. But the high false positive rate versus those that were detected, you just look at this cartoon of, I don't know where you go for your concerts, but this is the stage, and this is the balcony, and these are the seats. So low-dose CT will detect 3.5 people that screen in this whole audience, but the false positive rate is much higher. So that's a problem. And the problem is a financial problem. It's a problem of morbidity. It's a problem of anxiety. And this isn't only true of lung cancer in screening. It's true of mammograms, for example. There's a high frequency of false positive in mammograms. And my wife has had 4 or 5 now, all negative. She refuses to take any more because going through the biopsy and the psychology of all of it, she decided that she'd had enough. But that's between her and her physician. So low-dose CT now is funded by the U.S. government. So there's no reason that people who fit the criteria, and this is the current criteria, can't get low-dose CT. But there's about 10 million of those folks out there. And the capacity for doing low-dose CT is maybe 100,000 to 200,000. It's increasing. But Obamacare, Affordable Care, and also other Medicare will pay for it. So there is a need to identify high-risk individuals outside the criteria. And for example, never smokers. About 13% of the people coming into our clinic now are never smokers. Now, is that from passive smoking, or is it from parental exposure, or is it just bad luck in some way, or their job? So we need criteria to identify individuals and criteria and also prioritize these individuals into those who are greatest risk, so they don't have to stand in line for 6 months or a year or 2 or 3 years. As I said, at the time in 2010, there were about over 8 million. There are over 10 million now. So I'm about ready to win. So what's our strategy for early stage lung cancer and unmet needs? Well, one, of course, is to identify people who are at risk, and including those who are smokers or non-smokers, and for priority screening. And looking at various cytokines, urinary tumor metabolites is a quick assay, and also nasal epithelium is being explored, and CLIO-based assays are in progress. So you find someone that you decide, well, this person needs a scan, then you need to determine whether or not the lesion is a cancer or not. And as I was telling you earlier, most of them are not cancer. So you need early diagnostic markers, again, the serum cytokines, urinary tumor metabolites, and in the nasal epithelium, and there are other biomarkers that are being developed now. This is a big area, and I won't say any more about that than this. And then the next question is, well, it's cancer, but, and you do surgery on that person, and you remove that cancer, do they have metastases or not? And about 25–30% will have metastases, even though you can't see them. So the pathologist misses them, and they're micrometastases. So if you can identify those people, maybe you could offer them adjuvant therapies, either immunotherapy or chemotherapy. And one rationale might be that the micrometastases may be more susceptible than when you get a lesion that's 4 or 5 centimeters in size. And usually what kills these patients is, in fact, the recurrence of the original tumor. So there are several ways that people are focusing on this. So what was I preaching about? Stage 1 lung cancer, curative surgery, 25% recur and die of the disease. It can be stage 1A or 1B. So you have surgery in both. Some people are low risk and some people are high risk. So how do you identify those people who are at high risk? And precision medicine is the strategy that people are using now, including to decrease false positive rate, decrease financial costs, improve patient care, and guide further mechanistic studies. And these people you can observe to see if they change. These people, adjuvant therapy is what we would recommend. And these are just some of the fellows in the laboratory and some of the current people. Many of our collaborators, I've trained, I think, over 130 now postdocs. That's a lot. And, you know, they're chairmen of departments, they're deans of this, that, and the other. And so I'm very proud of all of them. And we get together every once in a while. There's like 60 of them in Japan, so we have parties and stuff, you know, all that kind of stuff. So I thank them, and I thank you for putting up with me after 5 o'clock. Happy to answer any questions. Yes. Yeah, what's your name? Joanna. Hi. You're on the second floor. Great. I hope it's not overheated like our lab is. Yeah, well, it goes from one to the other in building 37. Yeah. Okay, go ahead. What's your question?
I had a question about if you have a patient with a high mutational burden, if the lesions have kind of different mutational profiles, does that confuse the analysis of biomarkers? Or is that not?
So yeah, that's really a very interesting question. So people have been thinking about tumor mutational burden for various reasons, including finding that, well, maybe there'd be more neoantigens. So you'd have your immunotherapy might be more successful. It's controversial. There were initial results suggesting maybe that was going to be a case, but there are lots of results that don't find that to be the case. And tumor mutational burden versus biomarkers, that's not been well studied yet. And but that's something that can be done, including with the TCGA data or with data that we've collected. And that's actually something that we're quite interested in, and its relationship to the microbiome and metabolome. So that's a sophisticated question and a very good one. Thank you. Yeah.
Did I understand correctly that the relationship between these microbes that you studied, is it purely an information driving force, or is there another connection?
Okay. So there may be another connection. So some bacteria actually produce a protein that is a mutagen. And so that's something that we're searching for. In addition to Cetovorax, Klebsiella is also associated with smoking and lung cancer. And there's like 5 or 6 different species of Klebsiella in Cetovorax. So we're looking at each of those to see which might be. There's about a 60 base pair sequence that usually can tell you maybe this protein is there. And so that's a bioinformatic kind of example. And then you have to ask the question, can you find the protein? And then is there a way of inhibiting that protein? And one of our new collaborators has found a signature for that protein in one of these species that, like the signatures I was talking about with chemical carcinogens, there are going to be signatures associated with these proteins, small proteins produced by bacteria. And so that might be another clue to who gets cancer and who doesn't get cancer and what kind of cancer and so on and so forth. It's a very good question. Two very good questions. Want to try a third one? Want to go home? Okay, thank you.